Welcome to Human Physiology. Today, we are going to explore one of the most vital processes in the human body, the absorption of carbohydrates, proteins, and lipids. Imagine eating a delicious meal. How does that food actually get from your stomach into your bloodstream to power your cells? We will uncover the microscopic transport systems and the beautiful mathematics that make human life possible. Let us begin with carbohydrates, the primary fuel for the human engine. Think of your digestive tract as a crowded concert hall and the bloodstream as an exclusive VIP lounge. The massive crowd of complex carbohydrates cannot enter all at once. They must be broken down into individual fans, or simple sugars, like glucose, galactose, and fructose. This breakdown is critical because glucose is the absolute primary energy currency of your body. Without a steady supply, highly demanding organs like your brain simply cannot function, leading to severe fatigue and hypoglycemia. Now that we understand what these simple sugars are, let us look at how they actually cross into the body. They pass through specific security gates called transport proteins. Glucose and galactose are actively pulled into the intestinal cells against their concentration gradient by a transporter named SGLT1, which uses sodium as a copilot. Fructose, however, slips in passively through a different door called GLUT5. Once inside the cell, all three sugars exit through the bottom via a universal door known as GLUT2, finally entering the bloodstream. This biological transport system leads us to an important mathematical model. To understand how quickly sugars are absorbed, we use the Michaelis-Menten equation. Conceptually, this formula describes how absorption speeds up as more food is present, but eventually maxes out because there are only so many transporter doors. V is your current absorption rate. V max is the absolute speed limit when every transporter is busy. S is the amount of sugar in your gut. Km is a constant, showing the sugar level needed to reach half the maximum speed. As sugar concentration increases, the rate goes up, but eventually levels off at Vmax. Before we move on, let us see this formula in action with a concrete example. Suppose a patient eats a carbohydrate-rich meal, giving a gut sugar concentration of 20 millimolar. Their maximum absorption rate is 100, and their transporter constant is 5. We want to find their current absorption rate. We chose this formula because glucose relies on saturable transporters. We plug our values into the numerator, multiplying 100 by 20 to get 2,000. In the denominator, we add 5 and 20 to get 25. Dividing these gives us 80. Physically, this means the transporters are working at 80% of their absolute maximum capacity, rapidly pulling energy into the blood. Having calculated the absorption rate, let us visually reinforce what is happening at the cellular level. In this diagram, you are looking at a single intestinal cell, or enterocyte. At the top is the intestinal lumen, where the digested food sits. Notice the SGLT1 transporter actively pulling the yellow glucose molecule alongside a sodium ion. Right next to it, the GLUT5 channel allows the red fructose molecule to flow in passively. Finally, look at the bottom membrane. The GLUT2 transporter acts as the universal exit, funneling all these precious energy molecules directly into the bloodstream. This elegant microscopic system ensures no energy is wasted. Now that we have fueled the body with carbohydrates, let us look at how we harvest its building blocks, proteins. Think of a dietary protein like a massive, complex Lego castle that you just swallowed. Your body cannot use a fully built castle to repair skin or grow muscle. It must be completely dismantled into individual Lego blocks, or amino acids, to fit through the tiny mail slot of your intestinal lining. This is fundamentally important because every enzyme, antibody, and muscle fiber in your body is built from these absorbed amino acids. Without them, the body would break down its own muscle to survive. Understanding why we need these building blocks, let us explore how they enter our cells. Unlike carbohydrates, which must be single units, proteins can be absorbed as single amino acids, or small chains of 2 or 3. Single amino acids use sodium-dependent transporters, very similar to glucose. However, the small chains use a highly efficient transporter called pepT1, which cleverly uses a hydrogen ion gradient instead of sodium. Once these small chains are inside the cell, special enzymes called peptidases break them apart into single amino acids before they exit into the bloodstream. This dual pathway system ensures rapid and efficient harvesting of nutrients. To understand how the physical structure of the gut maximizes this absorption, we rely on Fick's law of diffusion. Conceptually, this formula explains how passive absorption depends on the physical properties of the gut. J is the total flux, or how much nutrient is actually absorbed. P is the permeability, showing how easily a molecule passes through. A is the total surface area of the gut lining, which is massive due to millions of tiny folds called villi. Finally, C1 minus C2 is the concentration gradient. If the surface area A decreases, the total absorption rate J will drop dramatically. Let us apply this formula to see how a healthy digestive system performs. We're given a permeability of 0.02, a massive, healthy surface area of 200 square meters, and a concentration gradient of 50. We want to calculate the total absorption flux. We use Fick's law to find the optimal scenario where the gut is fully utilized. We multiply the permeability, the surface area, and the gradient together. 0.02 times 200 times 50 gives us a final flux of 200 units. This high number physically represents a robust digestive system, easily meeting the body's demand for amino acids. Now that we have calculated the healthy baseline, we will mathematically prove why intestinal damage leads to severe malnutrition. We're setting up a comparison for a patient with severe celiac disease, which destroys the microscopic folds of the intestine. To isolate the effect of the surface area, we keep the permeability and the concentration gradient exactly the same as the healthy person. The only mathematical change is the surface area, which drops drastically from 200 down to just 50 square meters. This setup models the physical reality of a damaged, flattened intestinal lining. Continuing our derivation, we substitute these new values into Fick's law. Multiplying 0.02 by the reduced area of 50 and then by the gradient of 50 yields a total flux of only 50 units. When we compare this final result to the healthy flux of 200, we see the celiac patient absorbs only 1/4 of the nutrients, despite eating the exact same meal. This mathematical proof perfectly explains the intuition behind the disease. Without sufficient surface area, the body simply cannot sustain adequate absorption, leading to profound weight loss and nutrient deficiencies. Having secured our energy and building blocks, we now face a unique chemical challenge, the absorption of lipids, or fats. Have you ever tried to wash greasy hands with plain water? It does not work because oil and water do not mix. Your digestive system faces the exact same problem. To absorb oily fats into your water-based bloodstream, your body uses bile salts from the liver as a biological soap. This soap emulsifies large fat droplets into microscopic bubbles, allowing enzymes like pancreatic lipase to digest them. This complex process is vital, as fats make up your cell membranes and allow you to absorb essential vitamins. Because these digested fats are still afraid of water, they require a specialized shuttle system. They are packaged into tiny transport spheres called micelles, which ferry the fats right up to the edge of the intestinal cell, allowing them to diffuse across the membrane. But once inside, the body does something amazing. It rebuilds the fats back into triglycerides and packages them into massive protein-coated ships called chylomicrons. Unlike carbohydrates and proteins that go directly into the blood, these chylomicrons are exported into the lymphatic system first. This intricate shuttle system safely navigates fats through the watery environment of the body. To understand exactly why the liver goes through the trouble of producing bile to emulsify fats, we look at the mathematics of geometry. Digestive enzymes can only attack the outside surface of a fat droplet. This formula calculates the ratio of surface area to volume for a sphere. RSAV is the ratio, and R is the radius of the fat droplet. Conceptually, this tells us a profound biological truth. As the radius of the droplet decreases, the ratio increases dramatically. A higher ratio means more surface area is exposed for enzymes to work on, drastically speeding up digestion. Let us apply this formula to see what happens before emulsification. Imagine a person eats a heavy, fatty meal. Initially, the fat forms a large, single droplet in the stomach with a radius of 10 millimeters. We want to find its surface area to volume ratio. We use our geometric formula, substituting 10 for the radius. Dividing 3 by 10 gives us a final ratio of 0.3. Physically, this is a very low number. It means there is very little surface area available on the outside compared to the massive volume of fat hidden inside. Enzymes would take days to digest this. Now we will mathematically prove the power of bile salts. After passing into the small intestine, bile attacks the large droplet, breaking it into thousands of tiny droplets, each with a microscopic radius of 0.1 millimeters. Substituting this new radius into our equation, we divide 3 by 0.1, yielding a ratio of 30. When we compare this to our previous result of 0.3, we see the ratio is exactly 100 times greater. This proves that emulsification exposes 100 times more fat to digestive enzymes, turning a process that would take days into mere minutes. Let us visualize this entire lipid shuttle system. On the left side of the diagram, you can see the large yellow fat droplet being attacked by bile salts, breaking it down into those tiny, manageable micelles. Notice how the micelles ferry the fatty acids right to the border of the enterocyte, allowing them to passively diffuse inside. Once inside the cell, look at how they are repackaged into that large structure called a chylomicron. Finally, follow the arrow on the right. The chylomicron exits the cell and enters the green lymphatic vessel, or lacteal, safely bypassing the watery bloodstream until it is ready. We have covered the full journey of how our life-sustaining systems operate. First, we saw how carbohydrates are broken down into simple sugars and actively transported to fuel our brain and muscles. Second, we learned that proteins are dismantled into amino acids, and we proved how crucial the massive surface area of the gut is for absorbing these building blocks. Finally, we explored the complex lipid shuttle system, proving how bile salts maximize surface area for rapid digestion. You now understand not just what happens in the gut, but the beautiful physical and mathematical principles that keep us alive.